Okay, hello and welcome back to the deep dive. Let's imagine something kind of wild for a second. Think about plants way back when they first started colonizing land. They figured out how to put roots in the ground for water and nutrients and leads up in the air for sunlight and CO2. But as they got taller, sometimes, taller than buildings, how do they possibly get water all the way up to the highest leaves?
Yeah, and maybe even harder.
And even crazier, how do they get the sugary food they made in those leaves all the way back down to the roots hidden in the soil, which can't make their own food? It's like a logistical nightmare.
It's an incredible evolutionary challenge they faced, and their solution is nothing short of genius. A sophisticated internal transport and communication system.
And today we're doing a deep dive into one half of that system, the phloem. We're talking about phloem translocation, the process plants use to move those precious sugars made during photosynthesis, plus a whole host of other vital substances over long distances throughout their entire body.
That's right. This deep dive draws heavily from a key textbook in the field, Plant Physiology and Development. We're pulling out the essential nuggets, helping you cut through the complexity to grasp the core ideas.
So our mission for you today is to unpack the anatomy of this system, understand the physics and molecular machinery driving the transport, look at some classic experimental proof, explore how plants load and unload the cargo, figure out how they decide where sugars go, and even touch on the fascinating discovery that this system is also used for signaling. We're going to navigate this plant highway together.
Let's start with a fundamental challenge. Plants needed roots for water and nutrients and shoots for light and CO2. As they grew larger, these essential functions became spatially separated. Diffusion alone, which is great over microscopic distances, is simply far too slow to move water hundreds of feet up or sugars many feet down to the roots.
Diffusion would take years to get sugar to a root, seriously. So they evolved vascular tissues, xylem and phloem.
We've talked about xylem before, the tissue mainly responsible for transporting water and minerals upwards from root to shoot. Dead pipes, basically.
But today, it's all about the phloem. Think of the phloem as the plant's food delivery service and communication network, too. It translocates photosynthetic products, primarily a sugar called sucrose, from where they are produced or stored. The sources, usually mature leaves.
Right, or maybe a storage root in its second year.
To where they are needed for growth, storage, or survival, the sinks, like roots developing leaves, fruits, or seeds.
And you hit on something crucial. It's not just sugar. The phloem sap is a complex soup containing amino acids, hormones, inorganic ions, and even surprising molecules like proteins and RNAs, which act as signals. All this travels via the phloem.
Our deep dive will focus mostly on flowering plants, angiosperms, because that's where the bulk of the research has been done. We'll briefly mention how gymnosperms, like conifers, compare.
So where do we find this phloem system within the plant body? Is it obvious?
It's typically found alongside the xylem, usually towards the outside. If you picture a young stem cross-section, the vascular bundles have the xylem inside and the phloem outside. In a tree with bark, the phloem is the inner layer of the bark. Some plants even have phloem on the inside of the xylem. It's interwoven throughout.
Okay, and the actual conducting cells, the tubes carrying the sap, are called sieve elements. In flowering plants, these are specifically sieve tube elements, and in gymnosperms, they are sieve cells. Different names, similar job.
But these conducting cells aren't working alone, right? They can't be.
Absolutely not. This is key. Closely associated with sieve elements, especially in angiosperms, are companion cells. They are absolutely essential functional partners. The phloem tissue also includes parenchyma cells for storage and transfer, and sometimes structural fibers for support.
Okay, how do scientists know for sure that the sieve elements are the ones carrying the sugars? Seems like a basic question, but how is it proven?
Well, classic experiments provided early proof. One is girdling, removing a ring of bark from a tree, which strips away the phloem but leaves the xylem intact. Water transport continues upwards just fine.
That's the sugars.
But sugar transport from the leaves down to the roots stops dead above the girdle, causing sugars to accumulate there, making it swell up.
That's powerful evidence. And with radioactive tracing, you mentioned that.
Yes, using radioactive carbon, like 14C and CO2, allowed scientists to feed radioactive sugar precursors to a leaf. Then they could track the path of the sugars produced during photosynthesis. The radioactive label was found specifically concentrated within the phloem sieve elements, confirming they are the transport channels.
Okay, let's talk about the sieve elements themselves. They are really strange, specialized living cells. I mean, really strange.
They are unique. During development, sieve 2 elements undergo this radical self-pruning process. It's kind of wild. They lose their nucleus. Gone. Their main vacuole membrane, the tonoplast, gone. Their ribosomes, Golgi apparatus, even parts of their cytoskeleton, all gone. They essentially hollow themselves out to make way for flow.
What do they keep then? Anything?
They keep their plasma membrane, which is crucial. They also keep some modified mitochondria, plastids, and smooth endoplasmic reticulum, and their cell walls are non-lignified, unlike the rigid, dead xylem cells, so they remain flexible.
So they're alive, but minimalist, which is totally unlike the dead xylem cells. Why is being alive important for phloem transport? Does it matter?
It's crucial because the leading model for how phloem works, the pressure flow model we'll get to, relies on living cells maintaining membrane integrity and generating turgor pressure. Dead cells can't do that.
Okay. The sieve elements connect to each other through specialized porous areas, right? To make a tube.
Yes. In flowering plants, these connecting areas on the end walls develop into distinct sieve plates, which have relatively large pores, sometimes up to 15 micrometers across. These plates connect individual sieve tube elements end to end, forming long sieve tubes.
So essentially forming open, continuous tubes, like plumbing.
That's the current understanding, yeah. But for a long time, there was debate because early microscopy samples often showed these pores plugged up with, well, slime, basically, which didn't seem compatible with rapid bulk flow.
What was going on there? Bad samples.
It's now believed those blockages were artifacts caused by the sudden pressure release when the tissue was cut and prepared for viewing. The cell contents would surge towards the cut end and clog the pores, like a panicked evacuation blocking the exits.
Ah, okay. So newer techniques avoid that.
Exactly. Newer techniques like rapid freezing or observing living tissue with careful microscopy often show the pores wide open in angiosperms. The highway's open.
Okay, so the highway's open. What about gymnosperms? You said they're different.
Gymnosperm sieve cells are different. They have sieve areas, which are porous, but they don't form distinct sieve plates with those large open pores like angiosperms. Their pores are narrower and appear less open, often seeming blocked by membranes from the smooth ER.
So maybe less efficient transport.
Possibly. This anatomical difference might mean their transport mechanism is less efficient, or maybe operates a bit differently. It's one of the areas where we still need more research.
Right. If these tubes are under high pressure carrying valuable sugars, what happens if they get damaged? Like a hungry aphid tapping in or just a physical wound?
That's a serious problem. You lose precious resources very quickly because of that high internal pressure. Plants have evolved clever mechanisms to seal off damaged sieve elements quickly, like really quickly.
How do they do that? What's the first response?
There's a fast, short-term fix using proteins and a slower, more permanent one using a polymer. The proteins are called P proteins, found in most angiosperm sieve elements, but not gymnosperms. If a sieve tube is punctured, the rapid drop in pressure causes these P proteins, which are normally dispersed, to surge towards the wound and accumulate on the sieve plate pores, plugging them like a rapid response patch.
That's neat, like a self-sealing tire, sort of.
Exactly. And there are even special versions in legumes, the bean family, called photorespirators. They're these crystalline protein bodies that can dispose incredibly fast upon sensing damage signals, like calcium influx, and they can block flow reversibly.
Wow. And the slower, long-term fix.
That's callus, a type of glucose polymer, a beta-1-chloro-3 glucan. When damage occurs, the plant rapidly synthesizes callus right at the damaged site and deposits it in and around the sieve pores. It can seal off the damaged area completely within, say, 20 minutes or so.
Is that reversible? Or is that sieve element just done for?
Yes, usually it's reversible. The plant has enzymes that can dissolve the callus later, if the damage is repaired, or if it was deposited temporarily due to stress, like cold temperatures, or for dormancy.
And this sealing mechanism must be part of the plant's defense, right? Against pests.
Absolutely. Against insects that feed on phloem sap, inducing callus depositions can block their access. It's a form of resistance. Of course, some insects have even evolved ways to counteract this, like secreting enzymes that break down the callus. It's a constant evolutionary arms race happening right there inside the plant.
Fascinating. Okay, let's shift to the companion cells. They seem essential since the sieve elements are so stripped down, almost like ghosts.
They are indispensable partners. Sieve tube elements and companion cells are typically sister cells, formed from the same cell division. They are intricately connected by numerous plasmodesmata. These are cytoplasmic channels that link their interiors directly.
So they can share everything easily. Molecules, signals.
Yes, they allow for rapid communication and transport of solutes between the two cell types. Companion cells act as the metabolic life support system for the sieve elements. They handle protein synthesis, provide ATP energy from their abundant mitochondria, basically doing all the housekeeping the sieve element gave up.
And crucially.
Crucially, they are also the primary entry point for sugars moving from the surrounding leaf cells into the phloem system during loading.
And there are different types of companion cells you mentioned.
Yes, especially in the minor veins of source leaves where loading happens, we see at least three main types. There are ordinary companion cells, which have chloroplasts and varied connections. Then there are transfer cells, which are really cool. They have these elaborate finger-like wall ingrowths that hugely increase the surface area of their plasma membrane. They look like convoluted mazes inside.
Why the increased surface area?
It's specialized for moving large amounts of solutes across the membrane, suggesting they're involved when transport enters the cell wall space, the alloplast. And then there are intermediary cells, which are connected by tons of plasmosmata to the surrounding bundle sheath cells, and seem built specifically for taking up sugars directly from those cells to the connections what we call symplastic loading.
So the companion cell type gives us a big hint about how sugars get into the phloem in that particular plant.
Exactly. Transfer cells strongly suggest an apoplastic loading route involving membrane transport. Intermediary cells point towards the symplastic loading route, staying within the cytoplasm. We'll definitely circle back to these loading mechanisms soon.
All right, we know the player's sieve elements, companion cells, and the basic anatomy. Now, the trafficking patterns. Where do the sugars actually flow? Is it always down?
No, phloem transport isn't dictated by gravity at all. It's entirely driven by the relationship between sources, where sugars are produced or stored in excess, and sinks, where they are needed or stored. It's all about supply and demand.
Sources are typically mature leaves, doing photosynthesis.
Yes, mature leaves are primary sources because they photosynthesize more sugar than they need for their own respiration and maintenance. But storage organs, like potato tubers or beet roots, can also become sources when they remobilize stored sugar to support new growth, like sprouting in the spring.
And sinks are anything that's growing or storing, or just respiring.
Right? Roots, developing leaves, which aren't photosynthesizing enough yet, flowers, fruits, seeds, storage tubers, or roots during their filling phase. Basically, any part of the plant that is non-photosynthetic or isn't producing enough sugar for its immediate needs.
So the overall pattern is from source to sink. Does a specific leaf always supply specific sinks? Like, does the top leaf feed the top fruit?
Generally, yes. There's often a preferential flow based on proximity and the plant's vascular connections its plumbing network. Upper leaves tend to supply upper sinks like developing fruits or the shoot tip, while lower leaves primarily supply the roots. But the system is highly interconnected and can be complex, changing with development.
Okay. What exactly is in this phloem sap besides water and the main sugar, sucrose?
Water is the main component, the solvent, but the dissolved cargo is incredibly diverse. Carbohydrates, mainly sucrose, are the most abundant, often at very high concentrations, think 0.3 to 0.9 molar. That's like sugary syrup levels. This high sugar concentration is really key to the whole process.
Why sucrose specifically, and not simpler sugars like glucose or fructose? You mentioned stability.
This is a great point. Sucrose and other common transport sugars like raffinose or stachyose, which are basically sucrose with galactose units tacked on, are non-reducing sugars. Unlike glucose or fructose, they don't have a reactive aldehyde or ketone group exposed. At the high concentrations found in phloem, reactive sugars could cause unwanted chemical reactions. Non-reducing sugars are much more stable for long-distance transport. Less sticky, you could say.
Makes sense. What else is in the sap besides sugar?
Amino acids are major components, especially for transporting nitrogen around the plant in an organic form. You'll also find inorganic ions like potassium, which is very abundant, and phosphate, hormones that regulate growth and development, auxin, gibberellins, cytokinins, abscisic acid, and even those surprising things. Proteins and RNAs, which we now know can act as long-distance signals.
How do we even collect the sap to analyze it? It sounds difficult, being under pressure and prone to sealing.
It is tricky because of the high pressure and those rapid sealing mechanisms. One method is wound exudation, just letting sap bleed from a cut, but that can be contaminated or diluted, and the pressure drop itself can cause artifacts. The cleverest method involves aphids.
Aphids.
Yeah. They insert their feeding tube, called a stylet, with incredible precision directly into a single sieve element. Because of the high turgor pressure inside, sap flows passively into the aphid. If you carefully anesthetize the aphid and cut off its body, leaving the stylet embedded, sap continues to exude from the severed stylet for hours sometimes. It gives you a relatively pure sample from that single cell. It's like a natural micropipette.
Gross, but very effective. Okay, we know it's moving. How fast does it move? We said diffusion is too slow.
The speed of transport is often measured as velocity, in meters per hour. Typical velocity is around 1 meter per hour, but can range quite a bit, maybe from zero point 0.3 to 1.5 meters per hour, depending on the planting conditions.
Is that fast compared to, say, diffusion over the same distance?
Oh, orders of magnitude faster.
Yeah.
Diffusion over a meter would take, well, practically forever for sugar molecules. This speed is a critical piece of evidence. It tells us that phloem transport isn't just passive diffusion cell to cell. It has to be a bulk flow or mass flow process where the whole solution is pushed along, like water through a pipe.
Which brings us to the central question. What is powering this rapid bulk flow? What's the engine?
The leading hypothesis, the one that explains most observations, is the pressure flow model, first proposed by Ernst Munch way back in the 1930s.
Okay, Munch's pressure flow. How does it work? In simple terms.
The core idea is actually pretty simple. Bulk flow of sap is driven by an osmotically generated pressure gradient between the source and the sink. Think of it like squeezing one end of a water balloon. The water flows towards the less squeezed end, high pressure to low pressure.
So the pressure is higher at the source. How is that generated?
Exactly. At the source, sugars are actively loaded into the sieve elements and companion cells. This makes the concentration of solutes inside these cells very high, meaning their solute potential becomes very negative. Water then moves into these phloem cells from the nearby xylem, where water potential is higher, via osmosis. This influx of water pushing against the cell walls builds up significant positive pressure, turgor pressure.
Okay, high pressure at the source due to sugar loading, drawing water in. Then at the sink, what happens there?
At the sink, sugars are unloaded from the sieve elements, either used for growth or stored. This removal of sugar reduces the solute concentration inside the phloem, making the solute potential less negative, and therefore increasing the water potential. Water then tends to move out of the sieve elements, perhaps back into the xylem or surrounding sink tissues, and this causes the turgor pressure at the sink end to decrease.
So high turgor pressure at the source, lower turgor pressure at the sink, and that pressure difference pushes the entire sap solution along the sieve tubes from source to sink.
Precisely. It's mass flow water and all the dissolved solutes move together down the pressure gradient. Critically, the energy for the system doesn't come from actively pumping the sap along the tubes, but from the energy-requiring processes of loading sugars at the source and unloading metabolism at the sink, which create and maintain that pressure gradient.
How well does this pressure flow model hold up to testing? What predictions does it make that we can check?
It makes a few key testable predictions. 1. Because it's bulk flow, you shouldn't see true bidirectional transport happening simultaneously within a single sieve element. Things can move up in one tube and down in an adjacent one, but not both ways in the same pipe at the same time.
And the evidence on that?
Studies using different radioactive tracers have found bidirectional transport in whole stems, or petioles, but careful analysis shows it's always in different sieve elements or vascular bundles, or maybe at different times if a tissue switches from sink to source, never in the same individual cell at the same time. Also, water and solutes have been shown to move at pretty much the same speed, which is consistent with mass flow, not diffusion.
Okay, prediction 1 seems solid. Another prediction.
The energy needed along the path itself should be relatively low. Energy is definitely required at the ends, loading and unloading, but the flow itself is passive, driven by pressure. So energy is just needed for maintenance, keeping the cells alive, and maybe retrieving any sugars that leak out.
Evidence for that. Low energy cost in the pathway.
Chilling experiments on herbaceous plants are quite revealing. If you cool a section of the stem, it drastically reduces respiration and ATP production in that zone. Phloem transport is inhibited initially, but often recovers and continues at a good rate, even while the stem segment remains chilled. This suggests the direct energy cost for movement in the pathway itself is small, at least in these plants.
Right.
Strong inhibitors like cyanide stop transport, but they also damage the cells and cause plugging, so that doesn't really test the mechanism itself.
Right. In prediction 3, the sieve tubes, and especially the sieve plate pores, must be relatively open, not clogged, to allow efficient bulk flow.
As we discussed, modern microscopy techniques like rapid freezing or confocal imaging of living cells generally show sieve plate pores and angiosperms are indeed often wide-open channels, which supports this prediction. However, it's fair to say some recent, very careful studies using fluorescent proteins do show P-protein structures forming filaments or networks within the sieve tube lumen, even in apparently undisturbed living cells. The exact nature and porosity of these structures and their impact on flow resistance is still being actively investigated. It might be more complex than just wide-open pipes.
Okay, so mostly open, but maybe some internal structure. And the final prediction, the pressure gradient itself.
Yeah, there should be a positive pressure gradient, higher turgor pressure at the source than the sink, and logically, the taller the plant, the longer the transport path, the larger the total pressure gradient needed to overcome the resistance of the pathway.
What does the evidence say about that? Can we measure the pressure?
Measuring these pressures directly is tough. Using those aphid stylettes connected to tiny pressure sensors is the best way. Measurements in herbaceous plants and small trees do generally show positive turgor pressures in the phloem, maybe around 0.7 to 1.5 megapascals, which is pretty high. And usually pressure is higher in sources than sinks.
But what about in tall trees? Is the gradient much bigger?
That's where it gets a bit puzzling. While calculated pressures needed in tall trees can be higher, say up to 2 MPS, the actual measured pressures aren't always proportionally much higher than in smaller plants in the way the simplest model might predict. And getting systematic, direct measurements of the gradient along a single, continuous sieve tube over meters in a tree is still a major technical challenge. So while much evidence strongly supports the pressure flow model as the best overall explanation for angiosperms, especially the core idea of mass flow driven by a pressure difference established by unloading some aspects, like the precise magnitude of gradients in large trees and the exact role of those proteins in the lumen, are still areas of active research and refinement. We don't have all the answers yet.
So the pressure flow model is the leading theory, our best working hypothesis. But there are still some details to iron out, especially in challenging cases like tall trees. Are there alternative ideas out there, or modifications?
There are models that tweak the pressure flow concept or propose additional factors might be involved. For instance, one idea, the high-pressure manifold model, suggests the main pressure generation is concentrated heavily in the source leaves. And maybe the major resistance to flow isn't the sieve tubes themselves, but the plasmodesmata connecting the phloem to the final sink cells. That could explain smaller gradients in the main pipes.
Any other ideas?
Other relay models propose that maybe there's some active transport or pressure boosting occurring along the pathway, perhaps in stages, which could help maintain pressure over very long distances, particularly in trees. But as we saw, evidence for significant energy expenditure along the path, especially in trees, is still debated and needs more solid proof. Mathematical modeling is also used to explore how different factors influence flow.
Okay, let's go back to the absolutely critical step that starts the pressure gradient. Phloem loading. How do sugars actually get into the sieve elements from the photosynthesizing cells in the leaf?
Right. Sugars, mainly sucrose, are made in the mesophyll cells, the main photosynthetic cells of the leaf. They then have to move a short distance, maybe just a few cell diameters, through other leaf cells to reach the minor veins, which contain the smallest branches of the phloem network, including the sieve elements and their companion cells. The process of phloem loading is specifically the transport of these sugars into the sieve element companion cell complex. This is absolutely fundamental because it's what concentrates the sugar and establishes the high solute concentration at the source end.
And this is super important for agriculture, for crop yields, right?
Hugely. Understanding loading and later unloading is key to potentially manipulating plants to direct more sugars to the parts we actually harvest, the grains, fruits, roots, whatever. It's a major target for crop improvement.
You mentioned different types of companion cells hinted at different loading strategies earlier. What are those main strategies?
Broadly speaking, there are two main pathways sugars can take to get into the phloem, symplastic and apoplastic. In symplastic loading, sugars move entirely within the cytoplasm, from cell to cell, through those plasmogous middle connections, all the way from the mesophyll into the sieve element companion cell complex, without ever crossing a membrane into the cell wall space.
Okay, staying inside the cell network. And apoplastic.
In apoplastic loading, sugars exit the symplast at some point near the phloem and enter the cell wall space, the apoplastic. Then they have to be actively transported across the plasma membrane into the sieve element, or, more commonly, the companion cell. So there's a membrane transport step involved.
Apoplastic loading sounds like it involves crossing a membrane against a concentration gradient sometimes. Does that require energy?
Yes, it's very often an active process requiring metabolic energy. Why? Because in many plants, especially herbaceous crops, sugars are concentrated in the sieve element companion cell complex to levels much higher than in the surrounding mesophyll cells. Moving sugars against this concentration gradient requires active transport, which burns ATP.
And the mechanism for that active transport.
This typically involves specific protein transporters embedded in the plasma membrane of the sieve element or companion cell. The most common type is a sucrose proton supporter. These proteins use the energy stored in a proton gradient, which is itself established by a proton pump, an ATPase, burning ATP to co-transport sucrose into the cell along with a proton. It's like using a revolving door powered by protons flowing back in.
Got it. Using a proton gradient, ultimately fueled by ATP, to power sucrose uptake from the cell wall space. Very specific. What about symplastic loading? How do you concentrate sugars or be selective if it's just diffusion through connections? Seems tricky.
That's the clever part, especially with the polymer trapping model, which seems to operate in some species like squash or melon that have those intermediary companion cells and transport larger sugars.
How does polymer trapping work?
Okay, so sucrose diffuses from the bundle sheath cells into the intermediary cells through abundant plasmodesmata. Inside the intermediary cells, enzymes link sucrose with other sugars, like galactose, to synthesize larger sugar molecules, raffinose and stachyose. Here's the trick. These larger polymers are too big to easily diffuse back through the narrower plasmodesmata into the bundle sheath cells, but they can diffuse forward through slightly larger plasmodesmata into the sieve elements, so they get trapped in the phloem complex.
That's a very cool biochemical trick, making the sugars too big to go backward.
It really is. This process effectively concentrates sugars in polymer form in the phloem complex and maintains a low sucrose concentration in the intermediary cell, which keeps sucrose diffusing in from the bundle sheath.
Clever. And is there another type of symplastic loading?
Yes, there's also what's called passive symplastic loading. This seems to be common in many trees and perhaps gymnosperms. These plants have abundant plasmodesmata connecting to the phloem, but they usually have ordinary companion cells and transport mainly sucrose, not those larger polymers.
How does that work without trapping or active membrane transport? How do they build pressure?
In this case, there doesn't seem to be an active concentration step right at the phloem border. Movement from the mesophyll into the phloem is thought to be driven by a simple concentration gradient maintained by high rates of photosynthesis in the mesophyll. To generate enough turgor pressure in the phloem for long-distance translocation, these plants simply maintain very high overall sugar concentrations throughout their source leaves. So the gradient exists, it's just established differently.
So different plants use different loading strategies, apoplastic, polymer trapping, passive symplastic, often correlated with the type of sugar they transport, the structure of their companion cells, and maybe even their growth form, like herbaceous versus woody.
Exactly. There are definite patterns. Apoplastic loaders are often herbaceous crops, transport sucrose, rely on active transporters, and actively concentrate sugars at the phloem boundary. Polymer trapping plants use larger sugars, have intermediary cells, and concentrate sugars via synthesis. Passive symplastic loaders are often trees, transport sucrose or sugar alcohols, rely on high overall source sugar levels, and loading is driven by diffusion down a gradient maintained by photosynthesis.
It's fascinating how diverse these strategies are, and you said many plants might use more than one, or switch.
Evidence is growing that flexibility might be quite common. Some plants seem to have multiple companion cell types, or can potentially utilize different pathways depending on conditions, perhaps allowing them to adapt better to environmental stresses.
Okay, sugars are loaded at the source, building pressure. Now, how do they get out at the sink? Phloem unloading. It's got to be just as important.
Absolutely. Phloem unloading is the first step in getting sugars out of the sieve elements and into the surrounding sink cells where they're needed. And just like loading, the pathway varies a lot depending on the sink type. Is it a growing root tip, a developing seed, a storage potato, and its developmental stage?
Can unloading also be symplastic, just moving through connections?
Yes. In many growing sinks, like young developing leaves or actively growing root tips, unloading and the subsequent short-distance transport into the final sink cells can be entirely symplastic, occurring through plasmodesmata. In these cases, the sink cells are rapidly using or storing the imported sugar, so the sugar concentration in those sink cells stays low. This maintains a favorable concentration gradient for sugars to move passively via diffusion from the high concentration phloem into the low concentration sink cytoplasm.
So the energy cost there is mainly in the sink's metabolism, not the unloading itself.
Pretty much. The energy is used for growth, respiration, or converting the sugar into storage forms, which keeps the concentration low and the diffusion going.
What about sinks that store a lot of sugar, like fruits or sugar beet roots, where the concentration inside the storage cells gets really high? Symplastic movement seems like it would stop.
Exactly. In sinks that accumulate very high sugar concentrations, like sugarcane stems or developing seeds, an apoplastic step in the unloading pathway is often necessary. Sugars exit the sieve element companion cell complex and move into the cell wall space at some point before being taken up by the storage cells. This apoplastic step allows the storage cells to accumulate sugars to very high levels, often requiring active transport into those final storage cells, working against a steep concentration gradient.
And transporters are involved here too, then, for uptake into the sink cells.
Yes, often specific sugar transporters located on the plasma membranes of the sink cells are needed to move sugars from the apoplast into their cytoplasm, especially when those cells are accumulating high sugar levels. Sometimes enzymes in the apoplast, like invertase, even break down the sucrose into glucose and fructose before they are taken up by the sink cells via different transporters.
Complex pathways at both ends. Let's talk briefly about a leaf transitioning from being a sink, importing sugar when young, to becoming a source, exporting sugar when mature. How does that switch happen?
It's a fascinating developmental process. Young leaves are definite sinks. They need imported sugar to fuel their rapid growth and expansion. As they mature, expand, and develop their photosynthetic machinery, they gradually switch over to becoming net exporters of sugar sources. This transition often happens gradually across the leaf, typically moving from the tip downwards towards the base.
Do they stop importing and start exporting at the exact same time, or is there overlap?
These seem to be largely separate events, controlled differently. Import generally stops when unloading from the major veins, which deliver sugars early on, ceases. This might involve changes like the closure or elimination of plasmodesmata connecting the phloem to surrounding cells. Export begins later when the minor veins, the ones involved in loading, fully mature and develop the capacity for phloem loading.
And that loading capacity depends on?
Crucially, it depends on the expression and proper localization of those sugar transporters, like the sucrose proton symporters we talked about, in the plasma membranes of the sieve element companion cell complex in the minor veins. The appearance and activity of these transporters are key signals for the initiation of export capacity. Studies tracking where these transporters are made show a pattern that mirrors the sink-to-source transition across the leaf.
That makes perfect sense. The machinery for active loading has to be fully built and switched on.
Precisely. Gene regulation, controlling the development of minor veins and the expression of these transporters, is central to this transition.
So we've got sugars made in sources, loaded into the phloem, transported via pressure flow, and unloaded at sinks. Now, how does the plant actually decide which sink gets how much sugar? Sinks must be competing, right?
They definitely are. This involves two related concepts: allocation and partitioning. Allocation refers to how a source cell, like a mesophyll cell, divides up the carbon it fixes during photosynthesis, how much is used immediately for respiration, how much is stored temporarily, often as starch, and how much is converted into transport sugars, like sucrose, for export.
Okay, allocation is local within the source cell. What's partitioning?
Partitioning is the bigger picture. It's how the transported sugar, the photosynthate moving in the phloem, is distributed among all the various competing sinks throughout the whole plant: roots, fruits, new leaves, storage organs, etc.
So allocation within the source affects how much sugar is available for partitioning among the sinks.
Right. And they are linked in other ways, too, because a sink's ability to attract sugar, we call this its sink strength, depends on both its size, how big it is, and its sink activity, how rapidly it takes up and uses or stores sugar. That sink activity relates back to metabolic processes within the sink, which is sort of like allocation within the sink.
It sounds like this needs to be incredibly tightly coordinated across the whole plant.
Absolutely. Source activity, how much sugar is made, and the total sink demand must be balanced for optimal growth. How the plant partitions carbon between, say, root growth and shoot growth is critical for acquiring resources like water and light efficiently.
How do different parts of the plant communicate to coordinate all this partitioning? How does a root signal it needs more sugar?
It's complex, involving multiple signals. Communication happens through physical signals, like changes in turgor pressure within the phloem, which might rapidly signal changes in demand downstream. And, critically, through chemical signals. Hormones are key players. Auxins, cytokinins, gibberellins, ABA, transported in the phloem or xylem, influencing things like sink development, fruit set, or source leaf aging and senescence.
And sugars themselves.
Yes. Sugars themselves also act as signals. The concentration of sucrose or glucose in source leaves or sink tissues can directly influence the expression of genes involved in photosynthesis, sugar transport, storage, and metabolism. It provides feedback on supply and demand. For example, if sugars build up too much in source leaves because zinc demand is low, this can trigger a signal to actually slow down photosynthesis.
A feedback loop. And you mentioned proteins and RNAs as signals earlier. That still seems pretty wild that they travel in the phloem.
This is one of the most exciting areas of research in recent decades. It turns out that beyond small molecules like hormones and sugars, the phloem transports a surprising variety of macromolecules, specific proteins, and even various types of RNA over long distances. And these can function as potent developmental or defensive signals.
Like carrying complex instructions or messages, not just fuel.
Exactly. We've known for a while that plant viruses hijack the phloem to move systemically as protein-RNA complexes. Now we know plants send their own specific proteins and RNAs through the phloem network as well. To qualify as a signal, such a molecule needs to travel in the phloem, exit the phloem in a target sink tissue, and then actually modify the function or development of the target cells.
And is there solid evidence for this actually happening?
Yes, absolutely. Studies using fluorescently tagged proteins expressed specifically in companion cells have shown these proteins moving into the sieve elements and then being transported, sometimes over long distances, to various sinks. Movement out of the phloem into surrounding sink tissues seems more selective, though.
Any famous examples?
The classic example is a protein called flowering locust T, or FT. This protein is synthesized in the leaves in response to day-length cues. It then enters the phloem, travels all the way up to the shoot apex, the growing tip, exits the phloem there, and acts as a key signal, telling the apex to switch from making leaves to making flowers. It's basically the mobile flowering signal, or florigen.
So the signal telling a plant to flower travels through the sugar pipes. That's truly amazing.
It really is. And RNA signals are also being discovered and characterized. Specific messenger RNAs, mRNAs, and small regulatory RNAs, like microRNAs involved in gene silencing, have been found in phloem sap and shown to travel long distances, often bound to specific proteins forming ribonucleoprotein complexes. Some of these mobile mRNAs have been experimentally shown to influence gene expression and even cause visible developmental changes in sink tissues after moving across graft unions between different plants. It proves they are carrying functional genetic information.
This adds a whole new dimension to how plants coordinate their complex lives. It's not just hormones and sugars anymore. And plasmosmata, those connections between cells, must be critically involved in regulating this signaling too.
Absolutely. Plasmosmata are central to this entire story. They are not just passive pores, they are dynamic channels involved in symplastic loading and unloading. The sieve plate pores themselves are highly modified plasmodesmata, and they play a key role in regulating the movement of these signaling macromolecules between cells.
Do they act like gates?
In a way, yes. Transport through them can be passive for small molecules, limited by a size exclusion limit, SEL. But the selective movement of larger molecules like the FT protein or viral movement proteins seems to require specific interactions with components of the plasmodesmata, suggesting these channels can be actively regulated or gated to allow passage of specific, larger cargo. They are controlled gateways for information flow, not just simple plumbing connections.
Wow. We've covered a tremendous amount of ground. From the basic problem of transporting resources in a land plant, through the specialized anatomy and function of the phloem's unique cells, like sieve elements and companion cells, the remarkable ways plants seal damaged pipes using P proteins and callus, the source-to-sink transport patterns, the diverse cargo carried in the sap, not just sugar, the impressive speed of flow, and the leading hypothesis for how it works, the pressure flow model, along with the evidence supporting and challenging it.
And then we dove into the different strategies plants use to load sugars into the phloem apoplastics and plastic polymer trapping, passive symplastic, how sinks import these sugars using varied unloading pathways, the fascinating transition a leaf makes from importing to exporting, how the plant allocates carbon locally and partitions it systemically among competing sinks, and finally, the truly exciting role of the phloem as a conduit for sophisticated long-distance protein and RNA signals that coordinate plant life.
We've really tried to touch on all the key physiological processes, the developmental stages of these unique cells, the molecular players like transporters and enzymes driving loading and influencing sink activity, seen how classic and modern experiments reveal the system's workings, and highlighted how understanding these concepts is critical for basic plant biology and has huge implications for crop productivity. We aim to cover the whole chapter's scope.
It's clear the system is incredibly dynamic, highly regulated, and far more complex than just simple pipes moving sugar water. There are still many fascinating questions being actively researched: the precise mechanisms in gymnosperms, the exact physical nature and impact of proteins within the sieve tubes, the full suite of signals being transported and decoding what they all do.
Yeah, the revelation that the phloem is a major highway for macromolecules signaling proteins and RNAs carrying complex information is truly changing how we think about plant communication and coordination.
It makes you step back and appreciate the sheer ingenuity packed inside a seemingly simple plant. The internal life is incredibly sophisticated.
It really does. So here's a final thought to leave you with. Considering this intricate network carrying fuel, building blocks, and complex information throughout the plant, what other hidden signals might be traveling through this internal highway? What complex conversations are happening inside the plant, between roots and leaves, flowers, and storage organs that we haven't even begun to decipher yet?
It's a compelling thought, isn't it? The deep dive into the plant's inner world certainly continues.
Hopefully, this deep dive has given you a much clearer picture of this vital plant function, phloem translocation, and maybe sparked some appreciation for why it's so much more than just moving sap. Thanks for joining us.